Okay, so let's get started. So hello everyone for the weekend. Thanks a lot for everyone coming join us the weekend special thank you to our today's speaker. So it's really my pleasure to welcome our today's speaker, Dr. Fan Liu from all the way from Germany. So Dr. Liu actually currently is a professor from Department of Structural Biology uh, from the Molecular Pharmacology and from Berlin. So I just to give a very brief introduction for Fan. So Fan actually completed her undergrad study in biology school of life science in Fudan University in China. And then afterwards, she continued to pursue her PhD training in biochemistry from Department of Molecular and Structural Biochemistry, North Carolina State University in USA. So after she complete her PhD training, she actually moved to Europe. So did her postdoc fellowship in biomolecular mass spectra and then proteomics department in Utrecht University in Netherlands. And then since 2017, she decided to stay in Europe and move to Berlin as a group leader in Molecular Pharmacology Institute. And then since 2020, she become a full professor in structure interactomics in the Charité University Berlin. So Fan's group is particularly interested in developing and applying tools to characterize the complexity of protein interactions within the cells, such as cell signaling and differentiation. Use different method of mass spectrometric technologies, in particular cross-linking mass spectral. Her group mainly aim to chart the protein interactomes of complex biological systems. I think we're gonna hear a lot of the new tools her group developed, and I think are gonna be very widely used and very useful for different biological study, obviously including our immunology. So there are different aspects her group interested in, mainly including the how to develop a high throughput protein interaction strategies, and then also her group tried to define the structural interactomes of organelles, synapse cells, and viruses. So I think Vine really have a very productive and then a lot of groundbreaking discoveries during her both postdoc study and obviously after she become independent. With these achievements, she get numerous awards, which I think I'm not going to list all of them, but a few examples, including the Young Investigator Awards from the European Proteomics Association and Spinoza Young Talent Prize, and also Rising Stars in Proteomics and Metabolomics, DGMS Awards from 2024, and same time this year, MCP Lectureship Award 2024. So I think without further ado, really appreciate for your joining us all the way from Germany. And I think fine currently in California, which is 6 a.m. in the morning. Really appreciate it. We are very much looking forward to talk. Please, thank you.
Yeah, thanks, Chuan, for the extremely nice introduction. I'm actually at San Francisco, so this is my Golden Bridge is my background, but it's also where I am right now. So as you heard from my the introduction, that I'm not an immunologist, so my background is biochemistry, mass spectrometry, and I'm more of a technical person that we use mass spectrometry technique to study proteomics and especially protein-protein interactions. So today my talk will be it will not be technical at all, so I will just spend roughly 5 minutes to 10 minutes explaining the technology in a very simple form, and then I will give you, depending on the time, give you 2 to 3 application cases to explain to you how we use the cross-linking mass spectrometry to look at protein-protein interactions in complex biological systems. So just relax, it will not be technical, but rather will be biological focused. As I said earlier today, our lab uses this technique called cross-linking mass spectrometry to look at protein interactions, and then to study this, as the title says, the interactomic profiling and structure biology. So to start with the introduction of cross-linking, this is a workflow, so you can see that we use a cross-linker. This is a small molecule up here that has two reactive groups at both sides of the linker, and this reactive group here is NHS ester, which react with the lysine side chains on the protein. So basically, one NHS react with one lysine side chain forms a covalent amide bond. So then we put this cross-linker into different biological systems, including purified proteins, or this is organelles such as mitochondria or a phagosome, or even the intact cell. So essentially, we add this linker, solubilize in DMSO, add to this different protein complexes, and then it forms a variety of crosslinks on the surface of the protein. So then we do a normal digestion. This is a typical mass spec step, and then this will digest the cross-linked proteins into peptides. And then what we're particularly looking for is the little guy here, we call them cross-link. Then there's one side of the linker attached to one lysine residue on one peptide, and then the other side of the linker attached to another lysine residue on the other peptide. And then we use mass spectrometry to identify which lysine linked to which lysine, and this gave us the information of a long list of lysine to lysine connectivities. So then this is how we probe protein-protein interactions. And then keep in mind this, the cross-linker is roughly 10 angstrom. So this is the linker length over here. That is the a carbon chain with a disulfide, with a sulfoxide group, and this is roughly 10 angstrom, and which is 1 nanometer. So this cross-linker captures lysine to lysine residues that within 1 nanometer distance. So if you've done experiments like BioID or APEX, you know that those experiments gave roughly 20 to 100 nanometer resolution. If you've done super resolution imaging, that gives you a resolution of also something like 20 nanometer to 300 nanometer distance. So you can think of 1 nanometer as like a really short distance in terms of proximity labeling. So then what we get from such experiment is usually is a very large interaction network, as you can see on the left side of this slide. And every node here is a protein. And then if you see a gray line between two connected nodes, and that's a protein-protein interaction. So then it looks really difficult, but it's not that easy. Oh, sorry, it's not that complicated to decipher. That's it. So every node is a protein, every line is a protein interaction, and captured by 1 nanometer distance of the cross-linker. So you can see this is one of the older interaction network we got from the synapse, and you see some of the known protein complexes such as AMPA receptor over here. And then this is probably, you know very well that it's just the chaperone targeting, the TCC complex, and then some other protein complexes in the system. And what is also important is, as I mentioned earlier, that cross-linker captures lysine to lysine connectivity. So the resolution of the interaction is actually on the domain or subdomain level, where you see this lysine residue on this purple domain of the CNT and 1 protein interacting with this domain over here from the item 23 protein. So then this, as you can imagine, this technique works really well, especially for long proteins, as you can think that we can pinpoint the amino acid residue involved in the interaction interface, and which can direct point mutagenesis to block or disrupt the protein-protein interactions. So what we can do with this technology to discover novel protein interactions, so this is straightforward. If you see a protein interaction that linked to a known protein complex, so this could be a new interaction that involves in similar functions as this known protein complex. And then it also helps us to structurally elucidate protein binding interfaces. And this is just because the linked residue, they are at the interaction interface. So we can use together with AlphaFold modeling to find out where the interaction happens. Okay, so I also quickly mentioned about like the other approximity labeling approach, like LAPEX Turbo ID or BioID. You might have used it before to look at protein interactions. So the biggest advantage of cross-linking is the short labeling radius. As you can see that the cross-linker DSSO we use has a labeling radius of 1 nanometer, while the other technologies, they have like a much larger labeling radius, indicating that cross-linking is quite is a very high resolution technology compared to the other proximity labeling approach. Okay, so then this is just one summary slides explaining that the technology development over the past many years. So as I said earlier that I'm a technical person, so I spent roughly 10 to 15 years developing the technology to higher sensitivity and higher throughput. And then when I started, that was 2015 or so, that you see that we could only identify roughly 2,000 crossings from complex biological systems. But if you think about 2,000 crossings, that's roughly 200 PPIs. If we calculate 10 crossings per PPI of protein-protein interaction, so that means we're just only scraping the surface of a system that nothing really, we don't see much. So then over the years, we developed this is eye mobility like separation, then this is a real-time library search, and this is a new cross-linker has a affinity tag, and this is a new software that's actually just got published in Nature Method. So we will see it online probably in a month or so. We're doing proofreading right now. So then you can see that over the past 10 years, we're just slowly developing the technology from having only 2,000 crossings to 50,000 crossings last year to nowadays we reached 200,000 crossings. That's roughly 20,000 PPIs from a biological system. So after many years of effort, we finally managed to, let's see, capture more like a good number of protein interactions from a system, and which finally allowed us to use it to study some interesting biological networks. So, I mean, I want to maybe give you, so this is one very short very short story about like a technical story about protein-protein interaction, how we look at protein-protein interactions more very robustly. So, I mean, I'm a technical person. So usually the biologists, they see my data, they usually ask me, what's your FDR? Like how much I should trust your data? Because I think when like biologists look at proteomic data, we give you a very long list of protein targets. And then one of the big questions is which one is the real interactors? You usually just have difficulties of pulling out the real interactors from a long list of the mass spectrometry target that we give you. So then I just want to emphasize here that the cross-linking mass spec is really very robust or reliable. And then here we made a ground truth data set to hopefully to show you the evidence. So then what we did here is, so we actually together with Epsi Biotechnology, it's a company in Germany as well. So then we purified 256 human proteins, like individual proteins. And then we just have the 256 proteins in individual tubes. And then we essentially mixed two proteins in pairs and in individual tubes, and then made around 600 ground truth PPIs in the Eppendorf tubes. And then we cross-linked the protein pairs in individual tubes and quench it. So then every protein pair we put in one protein, then we know with the interaction, and the protein pairs we never put into the same tube. It's not like it's going to be a false positive. So by doing this, we made a ground truth data set containing roughly 600 protein pairs or PPIs, and then we used it to develop a software called Scout. This is our software, which is going to be published online in the near future. And then this is also, we also compared like different softwares on the market and for their FDRs. And then as you can see here, the blue bar, that is like the true positive, that is a real interaction. And then the yellow bar, that's a false interaction. So then you see that a lot of the software, they give a very high FDR when you just search, when you just do a search, and then you tell the software to give me 1% FDR, but actually it gave you much higher FDR than 1%. And meanwhile, our pipeline, and together with our software scout, you can see that it is very reliable. If you tell the software to give you 1%, it is indeed give you 1% FDR. This emphasizes the importance to have a reliable or highly reliable cross-linking pipeline together with a software engine, and this will give you reliable PPIs from essentially we test on different biological systems and then gave us very reliable result. So then that here I just want to emphasize that it's important to have a high confidence and pipeline, also a software, and then this is a good starting point to dig into the biology. So for biological applications, we have roughly different biological systems set up in our lab, including we started with organelles such as mitochondria, but we're also moving to different organelles like lysosome, exosome, Golgi, endosome. And then we also do whole cell cross-linking, just try to build a cellular interactome from the whole cell. And then we look at synapse and the virion particle and the virus host interactions. And then I will see how many stories I will tell. So you see here that we are able to change this beautiful schematic drawing from biorender to equally beautiful interaction networks from different biological systems. And this is the depth we can reach so far. So then for synapse, we'll have roughly 5,000 PPIs. From mitochondria, 3,000 PPIs. From cellular interactome, that's roughly 10,000 PPIs. So depending on the complexity of this of the system, we are ranging a couple of thousands to tens of thousands of PPIs. So what we can do with the data. So I will start with a small story about mitochondria. So this in this story that we're trying to look at the spatial proteomics. So essentially where the protein are, where the proteins are in the system. So this is one of the biggest questions in the proteomic field. It's called spatial proteomics, where just people try to identify the subcellular localization of the proteins. Like if in the whole cell, let's say if the protein is on the ER, where the plasma membrane, where the nucleus, and upon stimulation, and where the proteins go. Like maybe a plasma membrane proteins get cleaved upon stimulation and translocate into the nucleus and then later on land up in the mitochondria. So this is a question we try to study here as where the proteins are in the biological process. And to start with, we looked at steady state. So essentially this is a mitochondria. I think you know it very well already. Mitochondria has two layers of membrane, outer membrane and inner membrane. Maybe I moved to here already. And then that separates mitochondria into different subcompartments. And then we start with the mitochondrial interaction network, which is the fuzzy ball on the right side, as I showed you from the last slide. And then to how do we look at the spatial information from such a fuzzy ball interaction network? So the idea is actually quite simple. So we started with a few proteins that we know the localization, and thanks to the previous study that a lot of mitochondrial localization proteins, the localization of them are known already. So we started a few proteins with no localization. We call them localization markers, and then we just expand the interaction network. Then we will find many other proteins sitting in very close proximity as the localization markers. So then we just naturally find many other proteins sitting or inside the same subcompartment of the mitochondria. And then we can also further expand the network. Then we'll see some lysine residues sitting in this compartment. It's called inner membrane space. And then we'll also see some other lysine residues sitting in the mitochondrial matrix. And those proteins are like transmembrane proteins, right? So then we just did this, looking, just starting with a few localization markers, expanding the network, and we could find many other proteins sitting within the same compartments. So you can see that, so the mitochondrial two membranes separates into three compartments, and there's a purple or pink lines. Those are the protein interactions at the cytosolic compartments, and then the yellow lines, those are the proteins inside of the inner mitochondrial inner membrane space, and then the proteins labeled as blue, and those are the interactions in the mitochondrial matrix. And then, so what is also, so then we find many other proteins, new mitochondrial proteins. So then the idea again is very simple. If a protein interact with a protein with no mitochondrial localization, and this unknown protein is likely to be a mitochondrial protein as well. So then here we have, we identified a mitochondrial protein called FAF2 over here. So you see FAF2 binds to some other mitochondrial outer membrane proteins, indicating FAF2 is also a mitochondrial protein. And then on the right side is just the validation, and we do a lot of validation. So this is STAT imaging, TOM20 as the mitochondrial marker, and then correctly as the ER marker. So you can see that FAF2 nicely co-localized with both TOM20 and correctly indicating it's both a ER protein and a mitochondrial protein. So this is a Pearson correlation showing that it nicely correlates with ER and has a better correlation with mitochondria. And then this is just another validation experiment I will not explain, but just to say that this is a conventional biochemical experiment, also shows the spatial localization of proteins. But as you can see, this is like a time consuming and difficult to do, but then with the cross-linking is rather much faster and easier. And then this is again a summary slide showing that we can determine the membrane topology of 22 outer membrane proteins and inner membrane proteins. And this is just from one experiment, we can see the topology of membrane proteins from essentially all the membrane proteins covered in our interaction network. So it's very powerful and fast. And then I think I think I'll just skip this one. And then, so that's the mitochondrial story that I showed you. We can use cross-linking to look at the spatial arrangement of the proteins in the interaction network. And then we move to like a much difficult or a larger biological system that is a whole cell. So then essentially we add the cross-linkers into cell, and then identified or probed such a large interaction network. So you can see the network covers many different compartments of the cell, plasma membrane classes over here, peroxisome clusters, cytosol, mitochondria classes showing in red, and then endosome, nucleus, Golgi, lysosome, ER. So it's quite a lot of different subcellular compartments being covered by cross-linking. And then I just want to quickly show you here, this is as an example that we're trying to also to do some integrative structure modeling with the structure biologist, and to build up some atomic resolution structure of large protein complexes. And this has been a really difficult task for the cryo-EM, cryo-ET people, because for the cryo-EM people, for the single particle people, they need to purify the protein complex. And then there are quite a lot of protein complexes, especially shown here. This is nuclear pore complex. And then the core subunits are easy to purify. But whenever we think about the flexible regions and the disordered regions, it's almost impossible to purify from the whole cell or to do it in vitro reconstitution. And then so then you can see here that right. So this is the core complex of the nuclear pore complex, and this has been resolved already thanks to the cryo-EM, because the core complex only contains the structured protein. So this is easy to purify, and you can see that the majority of the cross-links match to the distance restraints really well on this core complex. And if we use the 40 angstroms as the cutoff, here that we measured C alpha to C alpha distance of the two lysine residues. So we gave it like a 4 nanometer distance due to the flexibility of the lysine side chain. And then you can see here that most of the crossings match really well to the 40 angstrom cutoff of the cross-linker maximum distance. And then, however, the whole MPC complex, there's another two big subcomplex, which is the first one is the cytoplasmic filaments, and the second is the nuclear filaments. And this two area contains a large majority of disordered protein regions that's almost impossible to study by cryo-EM people. And then when they do cryo-ET, there's also a lot of difficulties, as they couldn't identify the identity of different protein subunits. So as you can see here, that we we use the cross-linking data from the nuclear filaments, and then use the fragmented-based structure modeling approaches, try to pull out the information from the assemblies of all these protein complexes. You can see all these flexible regions in the area, and then build a model using cross-linking AlphaFold modeling and together with AI-based fragment modeling. So I hope this paper will also be published soon in a couple of months as in revision right now. So then I think I will, let's see if I have time to talk about synapse. So this is some data that we have with the synapse. And then, so synapse has a presynaptic compartment and a postsynaptic compartment. And then what we're looking at is how the molecules, they assemble on the synapse and how they interact together. And then how the pre and the synapse, postsynapses nicely align with each other to allow efficient synaptic transmission. So, I mean, I think I have, so then what we usually do when we have such an interaction network, we just dig into the subnetwork to look at some interesting interaction, new interactors. So this is a synaptic vesicle cluster, and then you can see we find quite a lot of no-synaptic vesicle proteins like the ATPase, and then synaptogerrin, synaptoporin, and then synaptoglycoprotein, synapsin 1 and synapsin 2 or so. And then we also, I mean, if you are a neuroscientist, you might see some of the proteins, they're not known to bind to the synaptic vesicles in this slide already. So we're following up on 2 of the new interactors to the synaptic vesicle. And then this is just another sad story about the postsynaptic density area. So in the neuroscience field, people have been trying to build a protein interaction network of the postsynaptic density area for a long time, almost 2 decades already. But it was never really possible because the PSD area, this is an electrical dense area, and has tons of disorganized proteins that are almost impossible to purify. And then also immunoprecipitation, or IP, doesn't work in this area because again, the proteins are really nicely densely connected. So if you do pull down on one of the proteins in the PSD, you either wash everything away if you wash too hard, or you pull out everything if you don't wash too hard. So this is like a really long-lasting question in the field. And then to look at the protein interactions at the PSD, and we tried to build it up by cross-linking data. So then on the right side is a study from a group that they used in vitro reconstitution, actually purified one by one the core subunits of the PSD components, such as PID95, SYNGAP, and then try to build this up in vitro to see the binding domain to investigate the binding interfaces between the many proteins here. And then you can see on the right side that by cross-linking, we could already get such information from one single cross-linking experiments. So you see that very well the NMDA receptor as the C terminus of the green 2B interact with PSE95, and then the C terminus of PSE95 interacts roughly about the middle region of the SAPAP1 and SAPAP2, and then the SAPAP1 and AP2, the C terminus of these 2 proteins again interact with roughly some things like the PDZ domain here of the shank protein. So then you don't need to look into the details, but rather say, I just rather want to say a conclusion here that our interaction map actually aligns really well with this in vitro reconstitution study. And what is also very interesting is we could easily add many more core components of the PSD proteins that's not being covered by the in vitro study. And you can see that SEP97, SEP122 is PSD93, and then we can add these 3 proteins into the existing interaction network and find out where those 3 proteins bind into which domain of the other core components of the PSD. Okay, then okay, maybe I think I can also say a few words over here. So I mentioned that cross-linking can capture domain level residue connection. So it's a very nice method to predict protein interaction interfaces. So we did this in high throughput. So essentially we started with 3,000 PPIs. This is from like the HEC data set, and then we developed the pipeline to predict protein-protein interaction interface together with AlphaFold modeling. And then in this pipeline that we screened 3,000 PPIs, as I mentioned, and we were able to predict 156 SLIM, this is short linear motif binding domains. So on the right side, you can see that this is like about 20 to 30 amino acid stretches, and then nicely sitting at the interaction domain to another ordered proteins. And so what we're doing now is we're gonna do high throughput mutagenesis or deletion mutants to remove the 20 amino acids involved in the protein-protein interactions as we predicted, and we see if we could disrupt these protein interactions. And we're gonna do this in high throughput using APMS approaches. Okay, so let me see. So this is just one of the interactions. You can see this is one of the interactions that we predicted. So this is a protein called UO12, and there's another protein called MAPK8. That's a MAP kinase. And then AlphaFold predicted a D motif containing sequence mediates the protein-protein interaction here. And then we made a mutant, MAPK8 mutant, mutating the proline and the leucine to alanines. And then we did this APMS approaches to validate the protein-protein interaction. As you can see that the MAPK8 was lost upon the mutation of the D motif, indicating this is a correct prediction. Okay, so I think I will just take another 3 minutes or 5 minutes to explain what we did with in virology. So here that we're crossing the intact virion particles and to look at the spatial organization, arrangement of the virus proteins and the host proteins inside of the intact virion. And then we can also add cross-linkers into the intact infected cells to directly capture virus host interactions. And then, this is a summary slide of this Nature Microbiology paper. It's really a lot of information there. So then what I want to emphasize is we could actually get a lot of information from mass spectrometry, such as spatial organization by cross-linking. This I explained to you earlier, using the mitochondria as a showcase to know where the proteins are in the system, right? And then we can also get a pairwise interaction probability estimated by cross-linking. So essentially, if the two proteins interact, or if the two proteins they never interact, what is the proper interaction probability between two proteins? And then we can also get protein copy number. So how many proteins how many copies of proteins per virion in this case, but you can also think about this is applicable to any other biological systems. And then we also get available structures from PDB or AlphaFold models, and then together with our collaborator in coarse grain modeling, and we made a beautiful virion model using all the information obtained on the left side of the slide. And then here we also showed, so this is another like biological follow-up shows that we indeed find also interesting virus host interactions. So then we find a new interaction that PP1 is a host protein that is a phosphatase, is a serine threonine phosphatase 1 is a phosphatase, and then interacting with a viral protein called PP150. So then we essentially using this cross-linking information, we can do a point mutation again to PP150. So then we made, so this is a wild type PP150, and then we also made a silk and RVXF motif mutation of PP150. So you can see upon mutation that the mutated PP150 lost the interaction with the host protein PP1. So indicating that again, we can pinpoint interaction interface between these two proteins. And then to check the biological function of the interaction between PP1 and PP150, so then we did, this is a very simple infection study that checking the virus titer upon infection using the wild type PP1, wild type virus and the mutated a mutated virus. So you can see that with both low MOI and high MOI, the virus type titer dropped 20-fold. So this is log scale on the y-axis. So then if you look at the and then that's about 20-fold on the right side using low MOI, so indicating that this PP150 is an extremely important interaction that's required for efficient viral growth. Okay, so with that, I think I'm going to, I should have a I don't have I should have a summary slide. So I hope I convince you that cross-linking mass spec is just really powerful approach to probe protein interaction networks from large biological systems, and it can be used to study protein-protein interactions, discover novel protein interactions, and then to probe the binding interfaces. And then to also, together with integrated modeling, we can build protein structures or the structures of large or macromolecular assemblies, especially for disordered protein regions that's difficult to purify by biochemistry. So with that, I would like to give my acknowledgement. I've been very grateful to work with a group of talented scientists at my lab. As I think my group, my institute is called Leibniz-Forschungsinstitut of Molecular Pharmacology. Not many people are doing pharmacology. Actually, we do whatever we want. And then so we have a group of really excellent collaborators. And Ludo from the Charité, that's our very own collaborator. He's an expert in herpes simplex virus. And then Free University in Berlin, as Mohsen said here, he's the AI person that did this course grid modeling of the virus with us. And then a lot of structural biology people and imaging people, and then the funding resources on the right side. So then we're also looking for candidates, PhD and postdocs. If some students are in the audience interested in learning mass spectrometry and not afraid of looking at large data sets and wants to have early access of many new interactions we have in the data, then you can contact me later. And this is it. And thanks for listening. And I hope I didn't go too fast and you could grasp at least the power of the technology. And then you can think about how you can apply this to your biological questions. Yeah, thanks very much.